Hi y'all, this video is to help you understand genetic engineering a little bit better. I want to thank Mrs. Yagua for this note that I'm going to give to you all today. So we're going to start with this concept of selective breeding versus genetically modified organisms, also known as GMOs. So selective breeding is when you have two organisms and you mate them so that you have desired traits as an outcome. So an example is in corn. We have corn that has properties that get rid of bugs. We call it insecticides, but it's actually embedded in the DNA code. Certain dog breeds, like my dogs, Edison and Watson, they are Bernedoodles, so they're Bernese Mountain Dog Poodle mixes, and that's because my dogs are selectively bred to be a certain temperament, a certain weight, and non-shedding. This process it takes, timing-wise, it's a long time for it to work itself out. Versus genetically modified organisms are much shorter time period. You're altering the DNA of the organism for a desired trait. Whereas selective breeding, you are purposely mating certain individuals together over many generations to get your desired traits together. GMOs are a shorter time span, and you're altering that DNA of the organism for the desired trait. So, for example, we have our little fish over here we call glowfish. Okay, and these glowfish have a jellyfish gene, and it is the gene that causes the jellyfish to have fluorescence. So glowfish, this gene was embedded into this fish species, and they glow the way they do. So just to recap, the corn is over here, as you can see, and it's over time the corn became bigger, juicier, more acceptable by consumers, and this is how corn used to be, and this is what the new corn is. Also, it's got insect fighting in DNA, so basically when the insects eat it, the insects won't like it. So lots of new things, new different types of crops, animals, etc. that have been a product of selective breeding. So, genetic engineering techniques, one of them is called recombinant DNA. So, this means you are inserting foreign genes into an organism. So you need a couple things in order to make recombinant DNA. Recombinant means a collection of different DNA or in one place. Almost like recombining. The word recombining, recombinant, it's like the same thing. So first you need restriction enzymes. And if you need more help on what those are, you should go back to our DNA scissors lab. And that is going to be helpful to figure out what are those restriction enzymes, where do they come from. They're essentially our DNA scissors. Our target gene, we need a gene to target. You can't just cut up all the genes, right? So you need to have a target gene that you want to insert this foreign gene into. And you need a plasmid. So a plasmid is going to be a smaller circular piece of DNA. I'll go ahead and write that here. and it comes from bacteria. So you can see it here. Here is what a plasmid looks like. Down here at the bottom. And you can see that it is not the bacterial chromosome DNA, but it's just a smaller circular DNA that is found in bacteria. And what we can do is we can essentially cut into it, put something new inside it, and then allow it to grow and repopulate and have more of that recombinant DNA. It's kind of cool. So how do we make this recombinant DNA? Which, if you're wondering what is this recombinant DNA, it's like looking at this picture, it's this here. Recombinant DNA is this bluer side, that's the plasmid that I just told you about from bacteria. And then you have to insert a gene. And you can't just insert a gene, you have to cut it correctly, and you have to cut the plasmid correctly, and you got to put them in there. So do you see how the plasmid was cut by something? And it's got some sticky ends, and so we were able to put in this new DNA into it, and that's called recombinant DNA. So that's kind of cool. So we're going to cut it using the restriction enzyme. From bacteria with sticky end cuts. We cut the plasmid from the bacteria with the same restriction enzyme, the same pair of scissors. You need to cut it with the same pair of scissors because then it will be cut at the same base pairs and allow you to put in a gene from something else into this plasmid. You're going to seal them together using DNA ligase. DNA ligase is like the tape, the DNA tape, if we're going with our analogy. And then you insert this recombinant DNA back into the bacteria. So you can see it in the picture down below. This is why plasmids are also called vectors, because vectors accelerate the process and they go from 1 to the X. Last but not least, bacteria replicate and express the new gene. So as you can see, once again, let me go ahead and show you guys again here. You have a bacterial cell and you have a plasmid. You take the plasmid out and you cut it with a specific restriction enzyme, just like EcoR1, for example. You allow that plasmid to have sticky ends. Okay, you see them here, 1 and 2. You cut the DNA that you want to put inside this plasmid. So let's see the gene for human growth hormone. You're going to cut it with the same pair of scissors or restriction enzyme and insert it into this recombinant DNA. So now it's called recombinant DNA because you've used restriction enzymes to cut the DNA from the human hormone, says gene for human growth hormone, into this plasmid with the same pair of restriction enzymes. Recombinant DNA just essentially means like hybrid DNA. Then you're going to put it back in to the bacteria so that it can repopulate. Okay, so it says how we make abundant amounts of certain proteins. Insulin is an example, human growth hormone, etc. And then it says helpful video for you to watch. Okay, so here's another picture of how we use restriction enzymes and DNA ligase to create recombinant DNA. So go ahead and pause and watch the video here. And I'll see you on the flip side.